and I'm going to get right into it. So I'm going to start with a story. So why do we do what we do? Let me start by telling you about a 40-year-old gentleman. This is a gentleman who lives in Kansas. He's married. He's a T-ball coach for his 3 kids. One of the nicest guys you'll ever meet. He's married to an ICU specialist. He was diagnosed with large B-cell lymphoma. He went through multiple clinical trials, receiving CAR therapy, CD19-directed CAR therapy on a clinical trial. And despite those 4 prior lines, his lymphoma is progressing. It's in his bones and it hurts. And then he comes to Stanford. And so what he has is something called large B-cell lymphoma. And at the bottom of the left side of the slide, I'm showing you a normal lymph node. And I kind of geek out on this stuff. I actually love seeing this architecture. You can actually see all these kind of pot marks. That's actually where immune cells are proliferating in response to presumed pathogens or antigens. And this is the normal architecture, but you can see just next to it, the large B-cell lymphoma. You see a complete effacement of the lymphoma, of the normal architecture, and it's just a morass of large B cells, literally what the disease is named after. And this isn't normal. And David covered this, but what this patient received was CAR T-cell therapy to treat that large B-cell lymphoma. And the way it works is you remove blood from a patient, you sort the T-cells out, you send it for manufacturing, and as David pointed out, we'll talk a little bit about how we manufacture here at Stanford. That's great to work with Steve Feldman's group. And you inserted some pieces of genetic material that insert permanently into the genome. These patients become a permanently genetically modified organism. It's kind of cool. So after we expand the T cells out of the body, we infuse them back in, and then they go all over, even to immunoprivileged sites like the brain. And the goal is to eradicate the cancer. And this has been practice changing. Here's one of many pieces of data that have shown how this has changed our practice. So this is data from Zuma 7. This was a randomized trial that compared Axi-cel, a CD19-directed CAR therapy, against the standard of care in patients who had progressed or relapsed within 1 year. And what you can see is a statistical improvement, an 8% improvement at 4 years. That's 8 more people out of 100 living. That's a meaningful improvement. And I'll note in the standard of care arm, for those who progressed, 60% of them went on to get CAR therapy. Historically, this number was a bit lower, around 20 to 30%. So CARs have made an incredible improvement, particularly the FDA-approved ones, and I'm grateful to have them. However, if you progress after a CD19 CAR, the outcomes are poor. Here you can see the overall survival after CAR T-cell progression. The median is only 5.5 months. That's not great. And even if you look out to 24 months, 2 years later, we still only have less than 20% of people still alive. This is horrible. We've started to learn why patients progress, and we think approximately 30% of patients relapse because you lose the target. CD19 either is mutated or it's downregulated. So we also have all these other therapies. These are all FDA-approved therapies, and what you can see is the response is quite low. On average, the response to the next line of therapy is probably at best between 10 and 20% complete response rates. And that's not particularly durable. You can see after about 3 months, most patients have progressed after all alternative therapies. And this is the field, the state of the art when this trial started. This is not great. So even though CD19 is often lost, there are other tumor-associated antigens, including CD22. It's found on the majority of B-cell malignancies. It's importantly not found on other critical structures in the body. And you can see here that CD22 expression is found across many different lymphoma subtypes. And so the second one in is large B-cell lymphoma. So that's what this trial will focus on today. But you can also see next to it follicular lymphoma, hairy cell leukemia, mantle cell lymphoma, marginal zone lymphoma. All of these different lymphoma subtypes express CD22. So when we recruited Crystal Mackel, we had seen the work that she and her group had done at the NCI, and they had used a CD22-directed CAR therapy to treat children with leukemia. These were high-risk patients. They had been through other CARs, they had been through an allo transplant. And so when she arrived at Stanford, Lori Muffley and I approached her and asked, can we test this in patients who are adults? both in leukemia and in lymphoma. And she was very kind and it started a wonderful partnership. So this trial started to treat both adult patients with leukemia, and this trial initially was started by Lori Muffley. And she was very kind and let me partner with her, and I helped lead the lymphoma component. And once it was clear how well this was working, she was very kind and I actually took over. But this has been a partnership with Lori Muffley the entire time. She's wonderful to work with. So this is a trial that we developed. We took patients who had predominantly had CD19 refractory large B-cell lymphoma. We used a standard manufacturing approach, and for those who want the details, let me know. But for the sake of time, I'll kind of say it's very important. But we used a closed system, a little bit different than was used at the NCI. We used standard lymphodepleting chemotherapy using fludarabine and cyclophosphamide, and we didn't know the optimal dose. So we actually designed a trial that would start with the known dose that was working in leukemia patients, and then if there was no dose-limiting toxicities, we would escalate to the higher dose. And then the follow-up period started with clinical assessments on day 28, and then 3 months, 6 months, and so forth. The primary endpoints were manufacturing feasibility. We had never really assessed this manufacturing approach before, so it was an important key component of this trial. We wanted to find the recommended phase 2 dose, and then we wanted to determine the initial safety and toxicity. And then the key secondary endpoints are your standard ones, mostly clinical outcomes. So overall response rate, duration of response, progression-free survival, overall survival. And then looking at other CAR T-cell associated toxicities. And then there were 3 correlative endpoints to try to understand the outcomes that we saw. So here is the fully enrolled patient disposition. So we enrolled 41 patients throughout a pandemic. So this was occurring between 2019 and 2022 was the last enroll date. And 1 patient withdrew, pursued alternative therapy, and 40 patients started manufacturing. 38 products were successfully made. The 2 patients who had their products unsuccessfully made were due to the T cells coming out of their body being very ineffective. We were basically unable to start the manufacturing process. And so we still consider that a manufacturing failure, but it wasn't because of the manufacturing. It was mostly patient characteristics. Patients were allowed to receive bridging therapy, and 14 patients did so. For those who had more than chemotherapy or radiation, namely chemotherapy, we ensured that they had measurable disease before we treated them with the CAR. Then patients underwent the standard lymphodepleting chemotherapy followed by a CAR therapy, and the median time from collecting the cells out of their body from apheresis to the infusion was 18 days, which is fairly rapid for our field. And here's the baseline demographics and disease characteristics of those enrolled. And what you'll see is these patients are very sick. 84% had elevated LDH. That's a marker of very aggressive lymphoma. On your Zuma trials, the prior trial that led to approval, was only about half the patients. So these were, the vast majority had elevated LDH. 97% had a prior CD19 CAR. Patients on average had 4 prior lines of therapy, and historically, after about 2 lines, cures are very rare. 29% had never achieved a complete response to any prior line of therapy. This including a few patients who had 8 prior lines without achieving a complete response. And I'll note that 34% of patients came from other academic centers during a pandemic to receive this trial, highlighting the role of Stanford within our larger community. And here's the data, and it's a busy slide, so I apologize for all the details, but for all how busy it is. But this shows patients' prior CAR, and the vast majority was axi-cel, which is the most common CAR given at the time. And as you can see in the upper right, is the progression-free survival is what you would expect for progressing patients. 90% of patients relapse within 6 months, which is when we expect patients to relapse. And there were a few later relapses, as you can see. And there were a few patients who had intervening therapy between the CD19 CAR and the CD22 CAR. And here's a list of all of them. I won't go through all the details. But what I'll note is the overall response rate to all of these other therapies was 16%. Complete response rates were only 10%. So not surprising, it's dismal. It's why we need this trial. And here are the key adverse events observed on this trial. We looked at rates of cytokine release syndrome. It's a flu-like illness that patients commonly get. What we saw was it was about 95% of patients on this trial had some form of cytokine release syndrome. What we really care about is those who have grade 3 or higher. And we only saw one patient at the higher dose develop grade 3 events. We also noted that even grade 2 events were more common at this higher dose. Neurologic events, or ICANS, is commonly seen after CD19 CAR. It actually leads to a lot of morbidity. It leads to patients being in the hospital for prolonged periods of time, requiring a month or two of rehab, particularly for folks in their 60s, 70s, and 80s. As we get older, it's harder. We saw very little. There were 4 patients who developed neurotox, and 3 of those patients had it for a single day, and 1 patient had it for 2 days. That gentleman who had it for 2 days had a history of, he was a ball player. He actually had a baseball that unfortunately hit him. He had some seizures, and so he was, it wasn't surprising he had a little bit of neurotox. But what was also surprising to us was the rates of what's called IECHS, immune effector cell hemophagocytic lymphohistiocytosis-like syndrome. It's a mouthful. This was not named when this trial started. In fact, the guidelines for this were just developed last year. So this was a new emerging toxicity that had not been fully described previously, and we saw it occurring in 1 in 3 patients at our higher dose. And at that higher dose, one of those patients actually developed an infection and passed away from a treatable infection, I would even argue. Because at the time, we really didn't know what this entity was. We didn't even know how to treat it. And so this has been a very humbling trial and sad trial at times because we've learned things while you're treating patients. The other thing I'll note in terms of toxicity is we've now seen 4 patients develop secondary myeloid cancers. And we think it's because they've had 4 prior lines, and this is on average their 5th. And every time you give a little bit of chemo or treatments, you're poisoning the rest of their body as well. And so we think that's an accumulation of just a lot of toxicity from basically poisoning our patients. So it also suggests that we need to give our most effective therapies first. But here is what was really shocking to all of us, and very delightfully shocking, which was patients responded. The overall response rate, which you can see here, was 68% of patients across both cohorts, and 53% had a complete response. Historically, you're talking about 10 to 20%. Even with the modern bispecifics, which came out after this trial, that's only about 35 to 40%. So at 53%, that's a remarkable outcome. And as I mentioned earlier, the overall survival was around 5.5 months. At the identified recommended phase 2, it's 25.7 months. We'll also note that the 3-year overall survival is 47%, which is nearly identical to ZUMA-1, the very first trial used to treat patients with a CD19 CAR. And out of the 20 patients who achieved a complete response, none of these patients have died of lymphoma. But unfortunately, patients have died. Some have died of their secondary cancers, 2 died from unrelated cardiac events, 1 died of infection related to this therapy. So that's why we actually lowered the dose. It was actually a little too toxic at the higher dose. And we didn't see any difference in overall response rates or CR rates or even PFS or OS. So we've identified the recommended phase 2 says the lower dose. It's a little more safe. But this is remarkable. Patients who achieve a CR have enjoyed very durable remissions, and we think are probably cured. But we still have work to do. This is looking at the secondary primary malignancy and other competing risks. And what you can see is less than half of patients die from their lymphoma. That's the blue line. But over time, you can see an accumulation of other non-relapse mortality, mostly from secondary malignancies. So this is something we're studying. We're studying patient recovery, long-term outcomes. Ann Kramer, who's sitting here in the audience in the green, will present our 3-year follow-up at ASH this year as an oral presentation. We're very proud of her. It'll be a nice presentation. What we also looked at is, were there subgroups that may benefit from this therapy, or is there subgroups that didn't do as well? So we're always keen to look at patients over 65, and what we found is no difference between those above and below 65. We also looked at those who had progressed rapidly after their primary CAR, so those who had relapsed within 3 months. And there was no difference between those who had progressed within 3 months or after 3 months from their prior CAR-19, and that's actually very surprising. A lot of us, including myself, thought that those who relapsed early probably were due to T-cell dysfunction and that we weren't going to be able to make an effective CAR subsequently. Luckily, I was wrong. Patients with elevated LDH, a marker of more aggressive cancer, historically poorer outcomes, did equally well to those with normal LDH. That's shown here. So overall, this worked across many, many different subgroups. There's relatively few patients, so there's an ongoing phase 2 trial, so we'll still take a look at all of these different subgroups, but we're excited to see that this is working across all these high-risk demographics. Let me share some more of the initial experience. These are the first 3 patients treated. And this is from a manuscript that John Baird and I put together and published in 2021, right after we had started this trial. So patient 1 had been through 5 prior lines of therapy. She had a large abdominal mass she nicknamed Lumpy. Lumpy was painful. Patient 2 was a patient referred from UCSF. She had been through 8 prior lines of therapy, had never achieved a complete response to any of those, including bispecifics, CAR, and pretty much had run out of options. Patient 3 had been through 2 prior CARs, been through axi-cel and through a trial investigating a CD19 CD20 CAR, which is looking quite promising, to be honest, but it actually lost CD19 expression. So in the last 2 patients, patients 2 and 3, were both high-grade B-cell lymphoma, having genetic abnormalities conferring high risk. So these were challenging patients, and they all responded. Lumpy went away by 6 months and never came back. Patient 2 had a mass that would grow out of her back, and if you had hugged her, it would fill the palm of your hand. That regressed. She's a nanny, almost 5 years. She's had a few coughs and colds from her kids, but has gotten through it. Patient 3, coaching football. I think winning too. Back to life. This is the goal we want for all our patients. And what I can show you is we actually want to know how and why these therapies work. And so this is, again, a busy slide. I'll direct your attention to the lower panel where it's showing expansion data for all the CARs. What you'll see is you can see expansion of the CD22 CAR. That's the more prominent in black. But you can also see the prior CAR-19. It's actually kind of hard to see, kind of on purpose. It's down here, down at the bottom, where you can see sort of the yellow. This CAR-22 seemed to expand about a log higher than the CAR-19. What we can also see, if you just follow patient 2 in green, is up in the upper right panel, you'll see ctDNA. And this is cancer DNA being shed into the bloodstream. And as the values drop, you're clearing the cancer. And what you can see is 2 of the patients clear their tumor by month 6, but patient 2 has ongoing reduction of their ctDNA. So what this suggests, if you combine the lower panel where you see persistent CAR expansion with the upper right panel, is that this CAR is active for at least 6 months. And these samples are still in the freezer for the later time points. And I have Rishi in the audience here whose project is to look at this. And I look forward to seeing when this truly clears. But this clearly shows that the CAR is active for at least 6 months. And so based on this phase 1 data, there is now a pivotal phase 2 trial that's ongoing. It's a multi-center study across 30 sites around the US. Stanford is leading enrollment on this, and we're very proud of our group for enrolling a lot of patients. And this is testing solely in CD19 refractory, so patients who've had prior CAR-19 and with large B-cell lymphoma. And we're more than halfway enrolled, and we hope to get a readout by next year. And also based on our data, we have now opened a new phase 1 trial here at Stanford in 6 new indications. Since this works so well in large B-cell lymphoma, we said, well, we'll work in follicular lymphoma. That's cohort 1 of this trial. How about mantle cell lymphoma, Burkitt's lymphoma, marginal zone lymphoma, and others? And so we have an ongoing phase 1 trial here at Stanford, and we're very excited about that. So what I've shared with you so far is somewhat the power of Stanford medicine. We have the ability to work in a team effort where we have experts in CAR T-cell manufacturing, cell pharmacists, correlative science experts, physicians and nurses, and team members that all work together to deliver excellent care. I'm amazed at our nurse coordinators for coordinating patients coming across all over the US. We even had referrals abroad. But that married dad, that T-ball coach, well, he's back to coaching T-ball again. He's thriving. He's in remission 3.5 years later. This CAR-22, now called pherocaptogene autoleucel, or Fira-cel, has been granted breakthrough designation by the FDA. That was obtained by Stanford. That's a very rare thing to have happen, which is an academic institution obtaining breakthrough designation. We're very proud of our Stanford team. And Fira-cel has been licensed to a commercial entity. As I mentioned, there's an ongoing phase 2 trial. We expect, hopefully, FDA approval by 2026, 2027, which is fairly rapid, about 8 years to go from phase 1 to approval, if that in fact happens. And I'm delighted to share with you that our phase 1 trial of the first 6 patients, 100% response rate. Of that, 5 are complete responses. So it's a very good start, a lot more work to do. So now I'm about halfway through, and I'm going to go from the clinic back to the bench. And I'm sharing with you here 6 people I think the world of. I've already mentioned some of their names. So this is John Baird, who was original fellow who helped lead this trial. We have Ann Kramer here in the audience, who's done just an incredible job of working with John and I to put the paper together, and was one of the lead writers of the paper. Is leading our single-cell RNA-seq data that I'll show you in a moment. Lily was a faculty member from Taiwan who came and worked with us for a couple of years, and has a paper resubmitted to Cancer Discovery, which I think will imminently get accepted. So she's done a great job, and I'll show you some of her work. Rishi's here in the front row here. He's led our work on the IECHS and cytokine analysis. And then Neha is my lab manager and senior scientist who's done a wonderful job of coordinating all of this and getting some of the science done. And then I have to just say thank you to Vita. She leads the CCSU group, who just makes all of this all the correlative work happen. But we want to look at these 4 different sort of bins: tumor characteristics, CAR-22 product analysis, how does the CAR expand and how does it impact clinical outcomes, and then cytokine analysis, and how does that all fit together? I'm going to share that with you now. So what I'm showing here is looking at expression of CD22 across our tumors. And we have 2 different methods. We have IHC, or immunohistochemistry. It's the method that's been around for probably 100 years. And an H-score is simply just calculating levels between 0 and 3 by how many percentage cells they have. So if your highest score you can get is 300. It means that all the cells are expressing it highly. But what we saw is patients below 200, that's in the red box here, that seemed to be a risk factor for progression, which makes sense. If you don't have the target, you're likely not to respond. Now, what was surprising is actually when we did this for CD19 CARs, CD19 expression did not predict outcomes. And that's been shown by our group previously. So again, it's small numbers, but if you take those 6, it is statistically different. Again, small numbers. There's a more modern approach using flow cytometry, where we can actually quantitate how many molecules are on the surface of the cell, something called quantibrite. And we largely think if you have less than 1,000 molecules, we think you're just not expressing the target. And if you look at those 4 patients who had more than 15% of the cells not having the CD22 target, all 4 of those patients progressed. Again, statistically different in small numbers. Then even at the time of relapse, we noticed that half the patients would have reduction in their CD22 target, again, suggesting this might be an escaping mechanism. But it's not so simple. We still had patients who had very high levels of expression. Some of these reductions were still above the average level. So we think there's other mechanisms that might explain these outcomes. So this is work put together by Ann. This is looking at CD22 CAR expansion. And not surprisingly, if you give a higher dose, you had a higher expansion. So that's shown on the left here. And it was about 3-fold higher. Not surprising, it was about 3-fold the dose. But when we looked at overall response rates, patients who expanded more had better outcomes. And so even independent of the dose. But that also came with higher grade CRS. So patients who had more expansion also had higher grade CRS, that flu-like illness. And this IECHS, this syndrome of immune system overactivation, those are some of our highest expanders. Neurotox didn't correlate, but we saw very little of it. So expansion matters. So T cells matter. And I think that's an important lesson here. So beyond antigen loss, T cell function is important. I'm going to go on a little tangent here because I mentioned this IECHS, and I showed you that they had very high expansion on the prior slide. But for the patients who developed this syndrome, we saw some clinical abnormalities that we can now recognize. So one is markedly elevated ferritin, very common with HLH. We see coagulopathy with the D-dimer rising very high and the fibrinogen plummeting. We see LDH elevated as we think immune cells are being engulfed by overactivated macrophages. We see triglycerides rise, which is also part of the HLH syndrome. We see cytopenias. We see liver dysfunction. This is a pattern that we've now learned to recognize. It's part of the guidelines. But we also went back to the lab and looked at the cytokines that were part of this. And this is, again, a very busy slide, but each column is a different cytokine, and each row is a different day. And at the top, we start on day 0, and the bottom is day 37. Unfortunately, this patient was in the hospital for 37 days. But we were able to collect a little bit of blood each of those days and then evaluate the cytokines. And what I want to highlight is over time, this patient is developing different toxicities. Initially, they develop CRS, and then that resolves. And then about day 15, they develop IECHS, a completely different entity. And what you can see is the pattern of cytokines change over time. You see one set of cytokines for CRS, and then it resolves, and then a different set of cytokines that occur during IECHS, and then as resolution. And at the very end, when she develops a bacterial infection, you see IL-8 go up, which is often common during an infection. So this just shows us that CRS and IECHS are two different entities, which have been debated in our field for a while. And overall, you can see that there are some genes, and it's very, I think, obvious in the far side in the red, the top rows are patients who develop IECHS, the bottom rows are patients who did not. Again, each column is a different cytokine, and then this is looking at various time points, day 7, 14, 21, 28, and looking at the fold change from baseline. And it's very apparent to us that you see all that red in the upper panels, and that's mostly interferon gamma-related cytokines, including interferon gamma itself. So we think overall, interferon gamma drives the overactivation, and we're still working on how exactly that interferon gamma is driven up to that level. But we think that's not only a signature of this, but also a target. We're now using drugs that block interferon gamma that are working to actually help ameliorate the toxicity. So this work is really important. And through all of the study, I was very fortunate to be part of this international panel to develop the guidelines of how to diagnose, grade, and treat IECHS. So it's been very important to me. I still remember one of our patients who died of this. So it's very near and dear to me, and I hope to one day not have that happen ever again. But let's turn our attentions back to the CAR-22, and this is work by Ann Kramer. We were able to bank material throughout the course of the trial. And so we have apheresis material, the CD22 CAR product, and then we took samples at day 14, which is the peak expansion, and then at day 28, which is a later time point. And over these 19 patients, we were able to get approximately 70 different samples to do single-cell RNA sequencing, which we can go cell by cell and ask what RNA is expressed there. And by looking at that pattern, we can identify correlates of response or toxicity, and that's what Ann has been working on. And so we took a representative group of patients. We largely chose half the patients to be responders and half to be non-responders. We wanted to include patients who developed IECHS and other high-grade toxicities. And we were able to generate all this data. It's nearly a terabyte of data. And here's a UMAP, and you can see how the different cells cluster. And so the material that's inside of patients, that's the peak, late, and apheresis material, all seem to cluster together. And then the product seems to cluster by itself separately. But using this UMAP, you can start interrogating questions like, how much of this prior CAR is left in these patients? The FDA was quite concerned that if you're genetically modifying cells and you take those cells and try to do that again, you're going to try to take already genetically modified cells and then cause cancer because you're integrating even more genomic material. So they were very concerned by this. So it's one of the questions we wanted to study. And what we can see is we can track the CD22 CAR very well. And about 500,000 of these cells are the are T cells, and about half of those are CAR-22 positive. But then we can ask those questions. And so this is, again, a busier slide, but we can ask, and you can see in the panel here, how many cells, individual cells, had both CD19 and CD22. And what I can tell you, out of about 500,000 cells, there's about 15. It's extremely rare. The prior CAR largely clears before patients undergo this second trial. And we even looked by early and late, and that's actually what you see on the left, is the patients in orange are those who relapse within 6 months. And we see detectable. It is above the level of quantification, but it is extremely low numbers. We're talking about even just the expression of CD19 is in about 40 cells. It's very, very few. It's actually a little bit higher. There's one patient that's 40. Everyone else is about 20 more. So it's a very rare event. You're talking about fractions of a percent. And when we looked at clinical outcomes, because some people ask me, well, actually, it might be an advantage. You're having a double targeting CAR at that point. That might be good. We see no benefit and no harm. So we don't see any. These are overall response rates. It was not statistically different based on their CR rate. We're looking at qPCR levels, those who are above the level of quantification to below, and we don't see any difference in PFS or overall survival. So it doesn't help, doesn't harm, and we even looked at toxicity, and we don't see differences in toxicity. Then we can go back to those cells and ask, each of those cells has a unique T-cell receptor. So can we track an individual cell over these 4 time points? And we can. So this is some data that says there are about 60 cells that are actually found across all 4 time points. And if you look at just 3 time points, there's a couple hundred. And so we can take a single cell and ask, what is the fate of that cell? What are the cells that are associated with response over time? What are the cells that are associated with non-response? And you can track the fate of each one of those. But we can also ask simple questions, and this work is ongoing by Anne, but one of the simple questions we ask is, if you have more diversity, so more different types of T cells, how do patients do? So that's what the Shannon Diversity Index is, and you can see in the middle panel that those who had more diversity had better outcomes. And that's been sort of corroborated by CD19 CAR data, but that's an important understanding of this product. We can also ask about different T cell subtypes. And there are what are called like naive cells. These are newly made T cells. There are central memory cells. And then as cells get more exposed to antigen, they become effector memory. And if they keep getting stimulated, they'll eventually become not angry, just disappointed T cells, and then disillusioned T cells, eventually exhausted T cells. We call them TEMRA. And if you have a high percentage of TEMRA, so if you look on the left here, those in purple, if you have a high percentage of those patients didn't do as well. You can look at the CR to PD ratios, and you can see that it's highly correlated with poor outcomes. The higher the number of TEMRA cells, the poorer the outcome. And that bore out also using a different method called flow cytometry. You can see that the outcomes were different. This is the PFS between the 2. So different subsets matter. And then we can start asking, are there individual genes associated with better or worse outcomes? And this is a volcano plot. And what it's asking is, are there genes associated statistically with achieving a complete response, or genes associated with progression of disease? And this is a method called pseudobulk analysis, acting as if the whole thing is like a mish put together. And there's a gene that stood out to us. It's a gene called June. This gene had already been characterized actually by Crystal Mackel. And so overexpressing c-Jun in patients in a CD22 CAR allowed for those cells to respond to lower antigen density and to persist longer. And even cleared CD22 positive tumors better in mice. And this wasn't overexpression. This was just found in patients who had a complete response. It's very satisfying. It looks like it's correlated. And has gone on to analyze and characterize a number of other genes. And for the sake of the general audience, I'll just briefly mention for the aficionados in the crowd. But the genes like LEFT1, FOXOs, these are genes that have been described previously by other groups for CAR19 and are seem to be associated with better product characteristics. And we're going to work on seeing their impact in CAR22. And there's another gene called FOXP3. It's a marker of Tregs. And we found that that gene was more highly expressed in patients who had progression. And work by Zena Good here at Stanford and a different group at Harvard have already shown that Tregs within your CAR product confer poor outcomes. So this work has been very fruitful. And my penultimate slide here shows, these are pathways that are upregulated in patients who achieve complete response, and not surprisingly involve pathways that regulate cell proliferation, interferon gamma, mTOR signaling, and so we have a better sense of how CARs get activated and confer a complete response. So in summary, what I'll show you is that greater expansion of CAR T cells correlate with better outcomes and increased toxicity. Having a high percentage of CD22 negative tumors appears to be correlated with progression. Patients with a higher proportion of terminal effector memory cells in products have poor outcomes. And we've identified a number of genes, including c-Jun, that are associated with achieving a complete response. And then genes enriched for interferon gamma, cell cycle, and proliferation are also associated with complete responses. We have a lot of work to do still, though. So some of the future directions that I'm interested in is, can we enrich the apheresis material with the right T cells? We know that some T cells don't contribute, in fact, may detract from your clinical responses. Cells like Tregs, TEMRAs. So can we sort the right ones and then manufacture the cells? We know that CD22 seems to be important, so if you have high expression, you seem to respond better. There are drugs that actually will upregulate CD22 expression. So there's a drug called bryostatin, and the question is, can you pre-treat patients, increasing their tumor CD22, and then have better outcomes? And then we also want to look at other tumor characteristics beyond CD22 expression. So are there mutations associated with poor outcomes? And I'm partnering with Ash Elizadeh and Rishi here in the audience to address that. And so we have samples from plasma where we're going to look by cell-free DNA to identify mutations and then hopefully determine resistance mechanisms by studying that cell-free DNA. So I'm going to conclude with just a lot of thank yous. All the folks on the slide are near and dear to me. It's been an honor to be part of this whole group. Our fearless leader, David Miklos, I appreciate you letting me have a job and letting me do what I do. Steve Feldman, his name is mentioned a few times. He leads our manufacturing group. That group does a phenomenal job. They have about a 96% overall success rate, which is just phenomenal. Lori Muffley, one of my colleagues here, my partner in crime on this trial. It was great to work with her. Beta, again, led our correlative sciences group. I'm grateful to her. Again, John, Ann, Lily, Rishi, it's been a pleasure to work with you guys. You guys are incredible. And of course, Crystal. She helped create this CAR, allowed Lori and I to study this in adults. She's been a great mentor, great collaborator. And then you can see here all of my physician colleagues, and it's been great to see them just be excited with us on this trial, and I'm grateful for all their care of our patients. And there's a number of names listed here. These are groups from the LCGM, from the laboratory space, regulatory affairs, clinical trial coordinators, pediatric group, pathology group, and then I'll mention my funding sources. So the Leukemia and Lymphoma Society, the ASH National Cancer Institute, the Marrow and Bob Seelig Research Fund. And so it's been a great group effort to get all of this done. And I thank you for your attention. And let me know if you have any questions or comments.
Thanks. I see a lot of questions here. Up this way. You showed that certain genes like Jun are associated with more effective responses, and Jun is also a proto-oncogene. So I'm wondering if you looked in in the patients who develop cancer to see if they have upregulated proto-oncogenes.
So we haven't looked at the tumor Jun expression as of yet. We have material left over, including a lot of the tumor blocks and cell-free DNA. It's not a very commonly mutated gene in lymphoma, so I'm not expecting it. But here, I think we're taking advantage of its function in T cells in its normal capacity without mutations or without abnormalities.
Hi, I'm Malali from Hematology. Thank you so much for that wonderful talk, and congratulations to you and all the group on the really fantastic work. So I had two questions. One was with the 11% rate of secondary MDS AML. Has that changed? Have you changed your practice in view of that? For example, do you screen for clonal mutations? Would it influence? I know there's competing risks because obviously these are very highly refractory patients. but has that changed your practice in terms of screening for patients who might be at risk of those outcomes? And then secondly, just to follow up on your point where you said, giving our best therapies early because of these concerns about secondary myeloid malignancies, do you ever envisage a scenario where you would sort of co-target CD19 and 22 concurrently in a CAR, or are there plans to do that?
Yeah, great questions. This data is still emerging, and I'm still worried about the patients who are still following. Part of the phase 2 trial, I highly recommend that we get bone marrow biopsies in patients to look for pre-existing abnormalities. And we're actually banking all that material to see how many existing CCAs or chip clones that are already existing, or if there's any abnormality predating. We didn't routinely require any assessments prior to this trial. So we don't know if they were pre-existing. Given that it took about a year for the first patient to develop a secondary malignancy, I don't think so, but I do highly suspect, based on Mark Hamilton's work, that there were already existing chip clones that just expanded post-CAR. And those are probably there, as you know, partly because of age. As we all age, we develop these abnormalities. But two, I think they also expand as we treat patients. As you repress the adaptive immune system, those clones will probably escape. And that's our lead hypothesis. Stay tuned to Mark Hamilton's work. He's going to present a talk regarding this, mostly around CD19 CAR, but he has all of our samples for the CAR 22. We're actually not only looking at the lymphoma tumor genetics, we're also looking for those other myeloid-related CCAS clones, chip clones as well, in the same cell-free material. So we're studying that. Remind me again, your second question was it? Co-targeting. Co-targeting is a great idea. We already have CD19, CD20 co-targeting CARs in our clinic. And stay tuned. I think I expect some, probably by the end of next year, some really promising data released. We're looking at our own data at Stanford, and I'm very excited to see how our patients have done, but these are multi-center trials, so we don't have the global data. But that's looking like a very promising approach. There are triples now in clinic. We're about to, we're hopefully going to have a triple opening next summer. So is it better to put it all in one cell, or is it better to infuse 2 bags of cells, one that targets 19, one? We don't know. This is open questions within our field. Both have been explored. I don't think we have a winner yet. But yeah, I wouldn't mind a double.
Thank you very much for your insightful talk. Here.
Yeah, thank you.
So you also showed data on the bispecific, and from what I understood is that you also get like 30 to 40% complete responses with the bispecific. How would you see treating the patients first with the bispecific, also in regard to the patients that might not yield good T cells, and then whenever the product is manufactured, that you give the T cells and hopefully they synergize?
Yeah. So bispecific antibodies for others is a way of using a molecule to link the tumor with the T cell. And what I have to say is I think all of these therapies, the bispecifics and CARs, need healthy T cells, and they don't play well together. So I worry that if you give a CAR first, that because of lymphodepleting chemotherapy, the remaining T cells are not very effective. So I find actually bispecifics immediately after CAR are not that effective. And there are people who are publishing on that as we speak. So I think if you wait something like at least 6 months before a bispecific works well.
I was more
reverse,
thinking
yeah,
of doing the bispecific, yeah. First.
You're talking about the reverse. And so the reverse, we also haven't tested because these are just emerging, assets within the clinical space. There was a group from France who showed data at ASH last year that said giving a bispecific prior to CAR seemed to not impact the CAR-19's outcome. Mm. It's retrospective, carefully selected. So we'll have to see. There is certainly preclinical data that suggests that when you use a bispecific, the T cells get exhausted. And then if you try to make a product out of those cells, I don't think they'll work as well. Mm. So I think the jury's still out on this. I kind of glossed over it because of the general audience of this, but in part of the phase 2 trials, there's a separate cohort specifically looking at bispecific exposed patients. Mm. So they've had both a bispecific and a CD19 CAR. I can't release the data, but I would prefer not to have a bispecific just before, generally speaking.
Okay, thank you.
I had two questions. One of them is, do you have any ideas about why you have a successful CD22 CAR in patients who have failed a CD19 CAR but maintained CD19 expression? And then the second one was for the apparently improved CD22 CAR expansion. It looks like it has the same co-stim domain. Do you have any ideas about why that's happening?
Yeah. So they have, so I'll answer the second one first, is the co-stim for axi-cell is a CD28, and this one uses a 4-1BB. Again, I glossed over that for the sake of the general audience today, but they are actually slightly different. So the expansion kinetics are a little bit different. The CAR19 expands a little bit faster, the axi-cell expands a little bit faster. So for the patients enrolled in this trial, it's not surprising to you that half the patients really didn't have much CD19 expression. So I think that largely was the reason why those patients didn't have a durable remission to that CAR. But for the half that did, there's still much we don't know. We don't know if they have mutations. We haven't assessed for that yet. We're going to look at it. So we're going to ask that question: is how many of those are mutated? But let's, for sake of argument, say they aren't mutated. I don't know. It's the real answer. We unfortunately don't have the prior products to study the T-cell fitness. We have 4 patients that we do, and so we're working on that now. So we're doing single-cell RNA-seq analysis, looking at the prior CAR19, and those patients actually have preserved CD19 expression on their tumor. So I think we'll learn something, at least in a small subset. So I don't know if it's that the T-cells recover because they had time between. There's a whole bunch of things I can speculate, but the answer is I really don't know.
On your volcano plot, I couldn't help noticing that next to June was FOSS, and I was wondering if you looked at, so heterodimer is much more efficient at activating the AP1 pathway. If you looked at whether it's actually heterodimer activity that more strongly predicted the response.
We haven't looked at it yet. This data is really hot off the presses, so I'm comfortable enough to share the data with you because we've looked at this multiple different ways by pseudobulk analysis, individual cell analysis. There's a whole bunch of methods. It's already been published by Crystal, but yeah, I don't know. I think it's a good question. I think that we agree it's the AP1 transcription factors probably collectively that probably matter. So we're planning to just overexpress June for now, but it might behoove us to do both.
Great. Moving on to maybe a little online for a moment. There's a few questions here. Dr. Ho asked, this is a great question. I was wondering, moving forward, will CAR22 be used earlier in the treatment course of lymphoma, or not only after they fail the multiple lines like 19 and 20?
I want to use what's best for patients. This is exciting data. I mean, we're thrilled to see how well this works after CAR19. We are opening yet a 4th cohort of that phase 2 trial in CAR-naive patients, so we'll see how well it works. But there's even a triple CAR that has it as part of the triple. There's already a trial now that has it part of a double that's being run out of Colorado. It's like CD19 and then this CAR combined. So I think there's going to be multiple strategies used to combine this CAR with others or give it in naive patients. We still see, not commonly, but rarely, some patients who are CD19, CD20 double negative, and that may be the patient population, if you have preserved 22, that you use this first. And what we're finding in the phase 2 trial, it's mostly 3rd line and 4th line patients, not 5th line like it was in this trial.
I'll also ask that Dr. Georgi asks, are there any ongoing trials on combining CAR T22 with other immunotherapies like checkpoint inhibitors?
We haven't pursued those trials yet. If you want to fund us, I'll glad to take your money and do that trial. We are this trial based on other trials. I'm not sure checkpoint would be the way I would go right away. I'm actually a little more interested in the double targeting or optimizing the products more. But those are sort of emerging questions in the field as well. There's other drugs like ibrutinib that people have speculated might enhance some of the T-cell function. So there's many of combos and all that's to be done. For now, I think the biggest bang for a buck is largely around double targeting and optimizing the product.
Another question online from Dr. Kudus. Are there any future application to adult patients with CML?
CML. And Mike, I'll find you later. He's next door to me in lab. And CML, I'm not sure what he's asking about CML there.
I'll take that as a no.
And like leave it. I would only have to speculate. I apologize.
No problem. If there's no other questions in here, I just want to also, Dr. Muffley had mentioned when you were giving her the kind acknowledgements that Matt, you're a wonderful partner too, and she said you did all the work.
Together with her.
Again, as I mentioned at the very beginning, this is amazing and the type of stuff that I'm just excited to be at Stanford, to be around work like this. So thank you for sharing it with us today. We're looking forward to maybe inviting you back in the future for updates as well. I think we will. And thanks everybody for being with us today. Have a great rest of the day.
Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.